Good afternoon, everyone, and welcome to another uh, uh, episode of this webinar we have bi-monthly. Um, Thank you for joining us today, and whether you're joining from near or far, we're thrilled to have you here for this exciting presentation. My name is Deb, and I'm your host, along with Dr. Jackson, and we have a truly engaging and insightful presentation lined up for you. We are confident you'll find the information shared today very valuable. As a reminder, this webinar is being recorded, so you can revisit the content anytime. Before we dive in, I'd like to cover a few housekeeping items to ensure you have the best experience possible today. First, if you have any questions throughout the presentation, please feel free, and our team will be monitoring it, and we'll do our best to address your inquiries. And we encourage you to actively participate. And if something resonates with you, please don't hesitate to share with your friends. Um, Now let's talk about what you can expect today. Today we are honored to have Dr. Adriana Silva, who's a postdoctoral research fellow with the Arizona School of Dental Health and Oral Hygiene. And she's here to discuss and describe the new SalivaEpi Biobank at ASDO and what we can do with it. So we're excited to get started. And without further delay, Dr. Silva, please take it over.
Thank you. Thank you, Deb. Thank you, Marie, for inviting me. And hi, everyone. I am Dr. Silva. I'm a postdoctoral research fellow here at ASDO. I have been here for a year, now a whole year now, and I'm very proud to join this team. During this time, we have been working several projects, and one of these projects is to establish a dental biobank at our university. And today I will talk exactly about that, the SalivaEpi Biobank, and how we can use this biobank to explore gene-environment interactions and dive research in oral and systemic health. Before I begin, I just want to share with you a little bit about how I felt in love with the field of genetic epidemiology. I was trained as a dentist and later completed both my master's and PhD in public health. This is my university in Brazil, located in the northeast region of the country. So basically, my formation has always been a blend of dentistry and epidemiology. And you might be wondering, where does the genetic thing come from? During my PhD, I had the opportunity to go beyond studying the epidemiology of oral cancer and dive into the genetic variants associated with the disease. This experience, I like to say that opened an entirely new world for me, and it was the moment I truly stepped into the field of genetic epidemiology, and I was hooked. By this time, I realized that oral diseases are not just clinical conditions we see and we treat in the dental chair. They are complex biological processes shaped by ancestry, environmental factors, behavioral, genetic susceptibility. So you have several things related with the occurrence of a disease. And from that point on, my research path changed completely. And that's basically what motivates me as a researcher. Today, we're going to talk about 4 topics. We're going to see the etiology of oral and systemic disease, the gene-environment interaction on the etiology of this disease. I'm going to talk a little bit about our ATSU SalivaEpi Biobank and the potential of this biobank and our current status of development. So let's start by the first topic, the etiology of oral and systemic disease. As clinicians and scientists, we have sought to understand the etiology of disease. It's what we always want to know, and based on that, to treat later the disease. Some diseases can be traced back by a single factor. That can be a genetic factor or environmental factor. Disease that can be traced by a genetic factor, generally we call the monogenic inheritance mode. It's the Mendelian inheritance that we have learned at the school. Basically, we have a specific genetic variant, a gene, that has a major effect, and because of that, this gene triggers the disease pathogenesis in the dominant, recessive models. An example is the sickle cell anemia. In this disease, we have a mutation in the HBB gene, and this mutation causes alteration in the hemoglobin proteins that assumes a typical structure, and because of that, we have some difficulties in oxygen levels and in blood flow. Other diseases can be traced by a single environment factor. We call environmental disease because this disease only occurs if the individuals are exposed to that factor, and when this factor disappears, the disease tends to stop. An example is asbestosis. We have generally when we inhaled the asbestos fibers, and it will cause some lung inflammation. But if you are not exposed to it, probably you won't have the disease. However, not all individuals who inherit a particular variant, a particular gene, or are exposed to a specific environment factor develop a disease. This small big point lead us to conclude that the vast majority of our disease and trait has a complex etiology, meaning that they happen by the interaction between genes and environment factors that will lead to a phenotype. Every time you hear the word phenotype, think on observational characteristic, a trait or disease, for example, the eye of your color, the eye of your hair, or I don't know, dental caries, a disease, a trait, a characteristic. So as I said, the vast majority of our diseases are not easy to study because they have a complex etiology. It's not related only for a genetic thing or environmental thing. We have multiple factors involved with the etiology of that disease. In most of the cases, the individual inherits a specific genetic variant. So you have several genes, and these genes have minor effects that combined, they can increase or decrease, they can shape the risk of a disease. We call it the monogenic inheritance mode, and this disease risk can be also modulated by environment factors. So here it's a hypothetical example. We have here that this trait is shaped by 4 genes. So the combination of the expression of these genes can lead to this trait. And it's why we have a range, and that's why we have a range of phenotypes in a population. Most of the people are in the middle, have a middle frequency here, but we also have rare cases of a disease or a trait in both extremes. One example is the height. It's why we have people, most of the people have a regular range height, but we have people that are very tall and people that are very small. Just another image, I really like this one. We can see that a disease, we have several genes, we call genetic variants that combined, they're gonna shape a disease risk, and they can interact with environment factors leading to the vast majority of our disease. Thinking on autoimmune disease, inflammatory disorders, chronic conditions, several kinds of cancers, and also the vast majority of our oral disease. Just to As an example, we have dental caries, the most common oral disease worldwide. We have several genes related with the occurrence of dental caries. For example, enamel formation, autoimmune response, genes for taste perceptions, for tissue repairs. So basically, the way that these genes express, we can have increased the risk for dental caries, our genetic risk for caries. But when certain genetic variants are combined with environment expositions, for example, presence of high dental plaque, poor saliva capacity, a diet rich in sugars, we can increase the risk of dental caries. And it's why some people has more caries than others, living same conditions or in different conditions, but having being being together. So it's because we have different ranges for the same disease. Another oral example is oral cancer. We have several genetic variants associated with oral cancer, and when they are combined with environment factors, especially tobacco, we can increase the risk of oral cancer. So now let's dive into a little bit more in the gene-environment interaction on the etiology of disease. We know that both genetic susceptibility and environmental expositions will contribute to human health and disease status. We have different theoretical models nowadays that try to understand, to explain how these factors occur together to shape oral health status. I really like this model, the integrated social environment model of health and well-being, because it tries to explain how social and environment factors combine via multiple ways to actually affect health and well-being over our lifespan. This is an illustration of this model. So basically here, it shows that individuals are exposed to different factors that contribute distinctly and collectively to shape our health. We have the environment factors that can be social, this blue layer. They include the global, cultural, and political factors that can influence the individual. We have physical factors such as geographic and climate conditions that can affect our health, and community level health factors such as social environment and building environment factors. These factors can also work together with the intra-individual factors such as lifestyle, genetics, biological characteristics, and this interplay between these factors can trigger behavioral and also biological responses influencing our health and maybe leading to some disease through all stages of life, including prenatal, childhood, and adulthood. In epidemiology, we have some models that try to explain gene-environment interactions, but before that, I really like to explain this term that is genotype, sorry. Genotype, we usually say that is the individual's genetic makeup that is determined by the DNA. So each gene has two alleles, and basically the way that these alleles happens, we call the genotype. So the genotype is our genetic code, and how these genes express and we see the characteristic is the phenotype that we already talked. So genotype is the genetic code, and phenotype the physical expression, the characteristic. Coming back for our models, we have 4 models that try to explain the gene-environment interaction. The first model is when you are exposed to certain condition, to exposition, and because you are exposed, you're gonna have the disease. You have a higher risk to have a disease, to have a trait. And if for any reason you have the genotype, a specific genotype, your risk for a disease is higher. However, if you are not exposed, even if you have this genotype, you won't have the disease. The second model is kind of the opposite. You have a specific genotype, and because you are genetic susceptible, you're gonna have a higher risk for the disease. But if you are exposed to a specific environment risk, your risk for a disease is even higher. And that's the same way, if you don't have the genotype, even though you are exposed, you are not in risk for the disease. The third model is when both the genotype and the exposition are needed for you have the risk for that disease, for that trait. And the fourth model is when both the genotype or the exposition can lead to the disease. You can have the disease because you are genetic susceptible or you are exposed to that. And when you have both, your risk is even higher. The gene-environment interaction studies have been going to study environment exposures interaction not only specific with genetic variations, as we saw, but with other biological layers. For example, the DNA methylation that we call epigenomics, the gene expressions such as RNAs and proteins that we call transcriptomics and proteomics. So we have different omics, we call the multi-omic layers. They are more complex to study, but at the same time, they try to bring a more comprehensive understanding of how genetic and environmental factors interactions in biological process contributing to disease and health status. Beyond all the advances we had in the study of gene-environment interactions, we still have a critical gap in understanding how this interaction contributes to the etiology and the pathogenesis of a disease, because to study gene-environment interactions, we need well-characterized data sets that integrate biospecimens and clinical, but also epidemiologic data. And usually the access that we have to biospecimens and structural patient data is limited and sometimes not easy. So because of these factors, we have some barriers in conducting translational research in epidemiology and genetics. Biobanks have been fundamental to advanced medical research because they work as a repository. They have biological samples that can be blood, tissue, saliva, combined with participant medical records. And these repositories allow us to easily identify biomarkers, genetic variants, environmental risk factors associated with the disease. We have some biobanks available, and especially for study systemic conditions, but we have few biobanks that include dental conditions and even fewer that integrate epidemiological and genetic data in a way that allow us to study oral and systemic disease. So basically, establishing a dental biobank at our university would provide an important source for us to accelerate research and discoveries in oral and systemic disease. And because of all these things, we decided to establish here a biobank. And now I'm going to present briefly the ATSU SalivaEpi Biobank. Our purpose is to establish a biobank in our university. Basically, we're gonna have biospecimens from saliva samples because they are very easy to collect and they are very resistible. We can store saliva for long periods without lose the capacity of extract DNA, for example. And Beyond the biospecimens, we're gonna have dental, clinical, and epidemiologic data to support ATSU students, residents, faculty, residents, scholars in conducting research, thinking on disease mechanisms, risk factors, treatment outcomes that can lead to a clinical application in the future. Initially, our biobank can support cross-section observational studies, which is studies that we perform with data collected just once in one time. The subjects for our biobank will be dental patients who seek treatment at our dental clinics. The recruitment will occur in the regular basis before they start the dental appointment, and they will they will join the study by informed consent that has already been approved by RAP. The data collection process is very simple. We're gonna invite the patient, and after approval, the consent, we're gonna ask the patient to donate a little amount of salivas, about 4 or 5 microliters, and this saliva will be stored in our lab for future analysis. Clinical, epidemiological, medical data will be extracted for the patient dental chart. Recently, the university changed the system for the ATSU system that allows us have access to a huge information of patients regarding several conditions, health conditions, dental conditions. So we have a really resource of information that can provide data and results later. And basically, with our biological specimens and clinical data, we're gonna create a data identifier data set using the REDCap. The saliva samples will be processed whenever they are needed for studies. The biobank now can perform DNA extraction and genotype procedures to support candidate gene association studies. These studies actually help us to identify which genotypes are more associated with a disease risk. So basically, we're gonna have our saliva sample stored. We're gonna extract the DNA. We can quantify to see the amount of DNA we have in each sample, and perform the genotype procedure that basically involves we perform the DNA amplification, that is multiply the copies of DNA we have to actually be able to read the allelic discriminations and identify the genotype. Clinical and epidemiologic data collected will be organized basically in 3 types of variables. We're gonna have variables related with dental phenotypes and treatments, such as dental caries, periodontal disease, restorations, endodontic treatment, whatever. Variables related with medical and oral health, and variables related with epidemiological characteristics such as socioeconomic, lifestyle, housing, and living environment. We're gonna share our data with studies that have been approved by our university. Clinical epidemiologic data and the genotype results will be exported from our dataset, containing only the sample size and the variables needed for perform that study, and we're gonna share in a PDF or Excel format. And now I'm gonna talk a little bit about the potential that the biobank has and our current status of development. This ATSU SalivaEpi biobank basically can support us with research, training, and collaboration. Thinking on research, now the biobank can support studies that try to investigate the salivary profile, and through simple tests such as saliva flow, viscoelasticity, and pH tests, Through these tests, we can investigate the risk of your oral and systemic disease. And we can also support candidate gene association studies that I briefly explained to investigate how genetic variants interact with environment factors to contribute to the risk of a disease, and also construct partial polygenic risk score panels based on specific genetic variants to predict a risk of a disease. We can also support some public health studies based on the population trends that we have in our data. And in the future, we hope that we can expand the biobank for the multiomic field. The multiomic field includes several omics such as genomics, transcriptomics, epigenomics, and basically each of these omics has your own biomarkers such as DNA, RNA, proteins, metabolites. Each one study one of these biomarkers and try to relate it to a risk of a disease. The biobank has also potential for training. Basically, it's going to work as a training platform for our students and faculty. HSU students and faculty involved with research will be allowed and welcome to shadow the laboratory activities. And because of that, they can promote skills in dental collection management, bioinformatics, interdisciplinary collaboration, and research. And we also hope that the biobank can contribute with collaboration, thinking both in across scientific collaborations and across institution collaboration. When I say across different disciplines, we can work together with clinical and basic science to work together to translate the molecular findings into clinical insights, which we call the translational research, and also across institutions, thinking both in national and international collaborations with researchers and internet educational institutions to create a framework that help enhance scientific, educational, and social impact for both institutions. And where we are, we started this year by creating the Biobank project. We prepared all the regulatory documentation, quality assurance material, all documents, forms, everything that we need for the biobank. We applied for IRB and got the approval in October. And now we are in the implementation process by coordinating the equipments, supplies, organize our infrastructure to starting the data collection by, I hope, the beginning of the next year. Just summarizing everything, I really hope that the biobank can actually create a repository of clinical, epidemiological, and genetic data to support our students and also increase the potential for translational research in oral and systemic health, can build foundations for predictive and preventive care, and can serve as a platform for training and collaboration. And by all these things, that the biobank can strengthen the HSU role as a leader in whole personal health. Thank you, everyone, and I'm available for any questions that you may have.
Thank you, Dr. Silva. Dr. Doublestein has a question in the comments. Considering interdisciplinary research efforts, how extensive will the intake medical history form be to gather other medical conditions like stroke, Parkinson's, breast cancer, lymphedema, low back pain, etc.?
We expect that we have all this information because, as I said, through Axiom, the system, we actually collect all the medical history of the patient, not all the dental, oral health, but all the systemic. So we know all the cardiovascular conditions, renal conditions, endocrinal conditions, all disease in all the systems we evaluate in our consult. So we have data in our chart. So basically, we have the information from the patient, and we can work together with our genetic variants to see associations between these systemic conditions and some genetic variants, but also to study associations between oral and systemic disease too. We hope we can advance to include systemic conditions too.
Dr. Tran has a question. What a cool resource for ATSU and the broader oral health research community. Do you have plans to provide the data on a website or submit the data to an existing genomic databases?
In the future, we expect to integrate any other repository to actually increase our data set. But now we are just in the beginning. We are starting to collect data in our university. It's a huge possibility, and we hope we can archive this point one day, for sure.
I have a question, Dr. Silva. What about risky behavioral data, risky behavior or diet, those data, like exposure kind of data can mediate?
Behavioral data, we have some. Diet data, not so much. I I can try to see because Exxon is very huge. I don't know if because I didn't find this part yet, but as far as I investigate in the system and I will organize the variables, we don't have too much information regarding nutrition and diet, but behavior we have.
Any other questions from the audience? Now is a good time.
I do. I have a question.
Hello.
Hi, Adriana. A wonderful,
Hello.
wonderful presentation. Many congratulations to you and Dr. Spalarich. This is a wonderful start. I think any university that has a biobank repository has so much potential. My personal experience has been that, these kind of, to detect biomarkers, you have to employ a lot of sophisticated analysis. I've had proteomics done, I've had lipidomics done, very costly. Now, my question to you is, what is the plan of action that you have in mind to ensure that, ATSU clinicians or clinical scientists or researchers get that kind of support that they're able to do good research? And at the same time, we also have a sort of a safety net for curbing the finances because we don't have our own innate research infrastructure. We don't have that kind of labs. I might be wrong. Maybe we do. I'm not aware of it. But I just want to know, like, what is at the back of your mind? Like, what is the plan of action there?
Sure, it's an important thing because all this study costs a lot of money. Now the Biobank, we actually have been founded, so we can equip our biobank to perform candidate-associated studies, as I explained. So we're gonna have the PCR, we can perform the real-time PCR here and actually study the genotypes. But for more omic analysis, as I said, we expect to have these equipments in a future based on others found that we archived. But for now, we're gonna have all the samples here. And for example, if a student wants to perform a study that it's beyond our capacity, we can work with facilities that perform, I don't know, proteomic analysis, and we can send the samples just for this analysis and come back. But for this part, the student has to be founded too. Here we're gonna support the students, the factory, everything for free. You thinking on our samples that we have and the equipment, but for example, supplies, the SNPs, these things, the study should contribute to this part. Okay, so I think it's actually important point, especially if you think on the omic field that all the science is coming to it nowadays. But for starting the studies we can support to candidate associate studies, for sure we have costs. We have to buy snippets, we have to buy supplies, but not a huge cost per analysis. So we can do that and work with facilities or the institutions to do omic omic studies in other layers that we are not in conditions in the moment.
Thank you.
You're welcome.
Any other questions from the audience? Because you're given a lot of food for thought, Dr. Silva.
Yeah,
So,
hope to talk another day about the advances in the biobank in the future.
yeah, absolutely. This is an ongoing discussion, isn't it?
Yeah.
Probably one of the most ambitious projects we've ever had. So that brings us to the end of today's webinar. And on behalf of ATSRI, I want to extend a huge thank you to our incredible speaker for sharing her insights and expertise on this matter. And of course, a big thank you to all of you for joining us, asking great questions and engaging in today's discussion. As a reminder, recordings are available on our YouTube channel, which I have copied pasted on the chat. We also have a Facebook and an Instagram handle, so please do check those out. And our next Research Nexus will be in the new year. This was the last offering for this year. And I believe, I think Dr. Silva did an amazing job with this being the last offering for the year. So we are going out with a bang. So I wish everybody a good, like, happy holidays, and we'll see you in the new year.